Welcome, friend. Welcome to our channel, Quality Guru. Friend, today our topic is contamination control strategy. It is basically a pathway for quality and safety. Friend, today we all are very much familiar with cleaning, contamination, sanitization, cross contamination. So it is we all are very much familiar. We are not new in this field. Introduction about CCS. Friend, today now all the drug manufacturers have employed CCS measure for decades as a core element of our GMP. CCS is basically a holistic approach. It demands tailored to the specific potential risk around each individual process. Many GMP practices are part of our company CCS, including, say for example, how our process and facility are designed, how our raw material and consumable are selected and managed, how our peoples are trained and developed. Friend, CCS is intended to drive continuous improvement and remediation. The success of CCS element is we need to understand how well our individual element work together, and thus we reduce the contamination hazard for a specific and defined process. Uh, it is a CCS is basically a multitude of GMP document. We are having numerous operational procedure and programs. We are having covered the risk assessment, validation program, technical document. Our CCS create umbrella documents that bring the relevant information together. It can be understood and evaluated holistically. Our CCS document should summarize the contamination control practices along with the underlying rationale and reference the supportive procedure and reports. We need to understand how to implement the effective CCS. For this, we need to follow and select best practices. We need to identify outdated practices, and we need to come up from old mindset. Reference guideline for CCS PDA technical report uh, number 90, contamination controls development in pharmaceutical manufacturing technical report provides guidance on how to establish an effective CCS for either a new or existing drug manufacturing facility or process. UGMP and XR1 require designing an effective contamination control strategy based on scientific assessment to understand the process and to apply effective risk management principle. The latest Annexure 2022 draft frequently uses the term risk, justify, and strategy that underline the importance of these concepts. Scope of CCS. Our CCS focus on contamination control practices against microbial and other advantageous agents, pyrogens such as endotoxin, foreign particulate matters in the manufacture of sterile drug, low bioburden drug substances, and as well as in the non-sterile drug that are vulnerable to contamination. CCS consideration: chemical contaminants include cross contamination and inherent particulate matter. Uh, CCS is a planned set of control for microorganisms, pyrogens, particulate, and derive the current product and process understanding that assure process performance and product quality. A CCS is a system that considered all the integral elements of pharmaceutical product manufacturing. Our CCS should cover microbial contamination, cleaning and disinfection, chemical contamination. Keep your CCS lean and simple. CCS contamination, that is identify the potential risk. Control, that is define our risk and prevent and do remediation program. Strategy, that is monitor and evaluate the risk. Step of CCS: identify your contamination source, define hazard critical control point, that is HACCP, process risk and its prevention. Build your strategy to monitor and control the risk. Prerequisite for CCS: process and technical knowledge. Define the entire process workflow, define HACCP, define risk and identify risk level, minimize the risk of contamination, access control, and monitor individually, define collective effectiveness, and follow our control measures. CCS for non-sterile drug product. Definitely, as CCS is very important for non-sterile, in the same manner, effective CCS is important for our non-sterile finished product. That are very susceptible to contamination. Microbial contamination lead to degradation of my product, loss of potency, and finally potentially unsafe level of microorganism and its byproduct being administered to our patient. So our non-sterile product and their intermediate should be handled and stored under the condition, the limit, the risk of microcontamination considering the specific hazard for microbial ingress and proliferation. Our liquid and semi-solid non-sterile products such as syrup, cream, ointment are inherently prone to contamination. It is because of due to the water activity level and support microbial growth. For these products, we are using some preservative are often added to avoid falling during the shelf life. Preservatives are rarely effective against all the potential contaminating species or high level of bioburden and may lose effectiveness over the product shelf life. So microbial control is also important to prevent and detect microorganisms during the process. Solid dosage products such as tablet and dry powder are generally less prone to contamination due to lower water activity. However, they are not contamination proof. Specific contamination hazards should be identified in manufacturing process and in handling and distribution of non-sterile products. Some process operations can lead unacceptable level of contamination, such as tablet coating, and there is potential for contamination during handling and dispensing activity. For example, blister pack protecting the product until administration. Foundation of contamination control strategy. For making a foundation of CCS, scientific knowledge, QRM, that is quality risk management, personal awareness, and quality culture is crucial to the development of effective CCS. Scientific knowledge, foundational elements. Scientific knowledge of process and technical knowledge of preventing contamination are the foundation of our CCS. Process knowledge. Potential ingress point for contamination to enter the process stream including particulates, microorganisms, viruses, bacteriophage, spores, endotoxin, and other microorganism byproducts. Say for example, endotoxin, proteases, and other metabolites. Potential proliferation point for microorganisms and viruses including bacteriophages to grow in the raw material, solution, process equipment, and process stream, thereby allowing formation of undesirable microbial byproduct. Microbial growth potential identified for each process step by assessing attributes such as pH, temperature, nutrients, water activity, and the duration of exposure, and or by performing a profiling study of process metrics such as the antimicrobial effectiveness test. Viral proliferation potential identified for each process step considering the presence of viral bacteriophage cell host. Process removal or reduction capability for potential contaminants. Say for example, filtration, heating, and chromatography. History of contamination event and trend. Definitely we need to know the history of contamination event and trend from microbial, viral, or from foreign particulate matter. Microbial and endotoxin concentration and microbial flora profile for existing process. Technical knowledge. Microbial attribute, behavior, and biofilm. For this, refer PDA Technical Report 69. Bioburden and film management in pharmaceutical manufacturing operation. Aseptic process PDA point to consider for aseptic process part 1 and part 2. Viral attribute, behavior, and host infection PDA Technical Report number 83. Virus contamination in the biomanufacturing, risk mitigation, preparedness, and responses. Process design principle PDA Technical Report number 41, division number 2008. For virus filtration, technical report number 42. Process validation of protein manufacturing, technical report number 44. For quality risk management, QRM, for aseptic process, technical report number 45. For filtration of liquid using cellulose based depth filter, technical report number 60. For process validation, a life cycle approach, technical report number 81. For cell based therapy control strategy, PDA control to consider for process aseptic processing part 1 and part 2. Facility and utility design principle, ISP baseline guide volume 3. For a sterile product manufacturing facility, PDA point to consider for ASIN facility, equipment design principle, PDA technical report number 34. Design and validation of isolator system for the manufacturing and testing of healthcare product, PDA point to consider for the aseptic process of pharmaceutical product in isolator. Cleaning, disinfection, decontamination, sanitization, sterilization principle, refer PDA technical report number 1, division 2007. Validation of moist heat sterilization process, cycle design, development, qualification and ongoing control, refer technical report number 3, division 2013. Validation of dry heat process used for depyrogenation and sterilization, before technical report number 26, revision 2008. Sterilizing filtration of liquid and technical report number 29, revision 2012. Point to considered for cleaning validation, before technical report number 48. Moist heat sterilizer system design, commissioning, operation, qualification and maintenance, before technical report number 49. Point to consider for biotechnology cleaning validation, technical report number 61. Steam in place, technical report number 70. Fundamental of cleaning and disinfection program for aseptic manufacturing facility, US Pharmacopoeia, Journal chapter 1072. Disinfectant and antiseptic, raw material quality, ICH quality guideline Q7A, good manufacturing practices, guidance for active pharmaceutical ingredient. Quality risk management principle, PDA report number 50. Implementation of QRM for pharmaceutical and biotechnology manufacturing operations, ICH quality guideline Q9. Quality risk management and foundation elements to achieve contamination control. The design of our new and existing manufacturing process and facility should employ QRM principle as to identify and access risk and intend to prevent and control contamination. The CCS should be designed with redundant individual control element so that no single failure in one element will result in contamination. For new process, existing contamination control technology and procedures that have proven effective in the process with similar risk profile can be used as a module. Say for example, connection, sanitization, sterilization practices, sampling method, new contamination control technology and procedures that are not yet proven effective in a relevant CCS should be designed and assessed using QRM principle to reduce the risk of contamination. New construction or renovation of production area are ideal opportunity for innovation. This should involve an evaluation of an existing CCS for any way to improve contamination control through innovative technologies such as closed or barrier system, automation and advanced monitoring and detection method. Personal awareness and quality culture foundational elements. The health of quality culture and the level of the personal awareness around contamination controls have a direct and significant impact on the success of CCS. Contamination control should be a priority that is reflected in the goal of the firm. Governance and oversight. A dedicated champion or team should oversee the performance of the CCS. The hallmark of a robust quality culture that promote contamination controls are focus on preventing contamination and their quality hazard, willingness to proactively invest time and resources to improve quality and prevent contamination, strong responses to contamination events that correct true root causes and prevent recurrence, management that appreciate and value the contribution and skill of employees, strong, supportive community of practices and network of subject matter experts who focus on continuous improvement, good cross department, good site communication, application of best practices and lesson learned. Employee who feel ownership over the quality of the product and process, including contamination prevention. Non-blaming culture. Employee who understand their responsibility to highlight quality risks and understand work toward solution. Thank you friends. Keep watching and give a positive response to us.